Today we are going to talk about cellular respiration. Cellular respiration is the way that we create all of our cellular energy, the cellular energy that our cells require for all cellular work. Today we are going to talk about cellular respiration. Cellular respiration is the way that we create all of our cellular energy that is required for cellular work. This cellular energy comes from the mitochondrion in the process of through cellular respiration. Today we are going to talk about cellular respiration. Cellular respiration is the way that all of our cells acquire the cellular energy they need to perform cellular work. This energy is made in the mitochondria. The mitochondrion is the powerhouse of the cell because he is able to take our organic food molecules and strip it of their energy to make our ATP. This ATP is then what we utilize to do all of our types of cellular work. The different types of cellular work are chemical work, mechanical work, transport work, electromagnetic work, electrical work, and bioluminescence work. Our cells require these different types of work in order to function properly. The driving force behind them all is our ATP molecule. ATP is generated through the process of catabolism. Catabolism is the way that our bodies break down food and extract all of the energy contained inside of them. This energy from catabolism is an exergonic reaction, meaning that it is spontaneous and occurs with no other added input of activity. This exergonic flow of energy from our food is enough to power the endergonic production of ATP. ATP then allows for the exergonic energy, this release of energy, to drive all of our cellular work or the process of anabolism. Cellular work is endergonic, which is energetically uphill, requiring tremendous amounts of energy. The ATP is there to give us that energy. But as you can see, once we perform that work, we are left with a lesser form of ATP in what we call ADP and inorganic phosphate. Once again, catabolism, energy stripped from our food, is enough to power the endergonic production of ATP. We see this in our energy flow throughout the life cycle. Energy comes from the sun. All of the sun provides us with all the energy we would ever require. Photosynthesis occurs inside of our plants. Plants take these inorganic molecules of carbon dioxide and water and actually convert it into organic molecules and release oxygen as a byproduct. Those organic molecules are then used for cellular respiration. Cellular respiration requires the oxygen as well as those organic molecules to produce ATP. Once this production of ATP is over, the end products, once again, are carbon dioxide and water, and the cycle continues. Remember that plants also require oxygen and organic molecules for their mitochondria. They have to go through this type of cellular work as well. Photosynthesis only provides them with the chemical energy needed to form the organic molecules, but their cells must do cellular work as well and require ATP made from the mitochondrion. If we look at the overall formulas for respiration and photosynthesis, we can see that they are almost exact opposites of each other. Cellular respiration takes glucose and oxygen and breaks it down into simple carbon dioxide and water molecules, whereas photosynthesis take these simple carbon dioxide and water molecules and builds these more complex glucose molecules and releases oxygen as a byproduct. cellular respiration is a catabolic reaction. It is exergonic in the release of all this energy. Photosynthesis is an endergonic reaction. It is anabolic because it stores energy in the form of the glucose molecule. Cellular respiration releases the energy from glucose to make ATP. Photosynthesis actually makes ATP from the energy found in the sun and uses that energy to provide the endergonic activity of producing glucose. Both respiration and photosynthesis require reactions called redox reactions. Redox reactions is a combination of two simple reactions. Recall back in Chapter 2 we talked about how ions are formed. Ions are electrically charged atoms: sodium ion, chlorine ion, calcium ion. These ions have either gained or lost an electron and have become charged. Redox reactions are very similar to the formation of ions. However, we are not talking about simple atoms, we are actually talking about molecules. Redox reactions are a combination, as I said, of two reactions: oxidation reactions and reduction reactions. In oxidation, a molecule will lose electrons. In reduction, this molecule will gain electrons. One way of remembering this is through the acronym oil-rig. Oil, oxidation is loss; rig, reduction is gain. Loss of electrons or gain of electrons. To look at this in a chemical reaction, we can see that they are coupled together. The X in this reaction has the electron and the Y does not. The Y wants the electron because it is extremely electronegative, and so it will do what it can in its power to remove the electrons away from X, and in turn causing X to be oxidized. Once X is oxidized, Y then becomes reduced because he has now gained that electron that was released. These reactions are always coupled together. When one loses an electron, another one gains an electron. We see this occurring throughout all of biochemistry. Because Y is reacting and is very electronegative and pulling the electrons away from X, Y is said to be an oxidizing agent. X in turn, since it is causing Y to be reduced, is what we call a reducing agent. If we look at the formula of respiration, we can see that oxygen is an extremely strong oxidizing agent because he is so electronegative and wants to pull the electrons away from our glucose. Glucose, on the other hand, is the reducing agent because it is losing those electrons and forming simple bonds of carbon dioxide. When we look at this overall formula of cellular respiration, we could see the oxygen being reduced to water and the glucose being reduced to CO2. What you also need to recall throughout this process is that the bulk of the energy is stored in those hydrogen bonds that those carbons are bonded to the hydrogens. Because of these carbon-hydrogen bonds, we can remove those electrons from the hydrogen and carry them on to oxygen. Well, how are these electrons carried? The coenzyme NAD+ is a good place to start. When we look at this NAD+, what we can see is that it is a dinucleotide. What you need to recall is that a nucleotide contains a nitrogenous base, a sugar, a 5-carbon sugar, and a phosphate group. Nicotinamide adenine dinucleotide has an adenine nucleotide, just like what we find in RNA, and it has a second nucleotide, a second phosphate, a second 5-carbon sugar, and then another nitrogenous base known as nicotinamide. When it is positively charged, this is the oxidized form of NAD, and so it is referred to as NAD plus or positively charged. NAD plus is a coenzyme. Recall that coenzymes are assistants to our enzymes. They are non-protein organic molecules. This image of NAD plus should show you that these are not amino acids. This is a non-protein organic molecule known as a coenzyme. NAD plus just happens to assist these enzymes and enzymatic activity by carrying hydrogens and electrons away from the food source and on its way to oxygen. So if we look at NAD plus and we combine it with 2 hydrogens from our food, remember all of the chemistry or all of that energy is stored in those carbon and hydrogen bonds of our hydrocarbon. So if we remove 2 hydrogens from a carbon, we are removing the 2 electrons that it has and the 2 protons. These 2 electrons and 2 protons then find their way through an enzyme known as dehydrogenase. This enzyme will then transfer 2 of those electrons to the NAD+, one here and one here. As you can see, that electron has gotten rid of the positive charge. One proton is also carried on the NAD. And in turn, we have the reduced form NADH. But there is still one proton left over, and that's kind of hanging out here on the end. So when we say that NAD plus is reduced, it is now reduced to NADH plus another proton, a hydrogen ion, or H plus. These electron shuttlers occur throughout cellular respiration. They allow for the electrons to travel from our food to oxygen. Remember, oxygen is extremely electronegative and all it wants are those electrons from our food sources. Because of this, it will pull the hydrogens away from our food. These NAD pluses act as shuttle systems. Just imagine him like being a taxi cab, picking up its fare and carrying it to its destination. When there's nobody in the cab, he's flashing vacancy, and when he has somebody inside the cab, his vacancy sign is turned off. There is another coenzyme that will come into play in cellular respiration as well, known as FAD. He too carries electrons away from food sources all the way to oxygen. Now let's look at an overview of aerobic respiration. There are 4 key components or parts to respiration. What we see here is that there is glycolysis, which is the splitting of glucose, the transition step, the citric acid cycle, and the electron transport chain. Throughout these 4 major steps, we will see glucose combining with oxygen to be broken down into carbon dioxide and water. And ultimately, our net gain, or our overall reason for going through this process, is to produce ATP. And one glucose molecule has the ability of creating 38 ATP molecules that will be the driving force of cellular respiration or cellular work. If we look at glycolysis, remember this glyco means sugar, lysis is splitting. So this is the splitting of our sugar molecule. What we see is that glucose is broken down into 2 pyruvates. Pyruvates are a 3-carbon sugar. Because glucose has 6 carbons to begin with, we end up with 2 3-carbon molecules at the end. In order for glycolysis to occur, however, it requires ATP. Just like if you're sitting at the top of a hill on your bicycle, you have to put in a little bit of energy to pick your feet up off of the ground and put them onto your pedals before you can start coasting down the hill. Glycolysis requires a little bit of energy to get everything started. Once we get started, glycolysis has the ability of producing 4 ATPs through a process known as substrate level phosphorylation. It will also generate 2 NADHs. Now remember, the NADHs are our coenzymes, and they are carrying electrons and hydrogens to oxygen. The first step of glycolysis occurs in what we call the cytosol. Glycolysis occurs outside of our mitochondrion. The key component of glycolysis is that it does not require the oxygen. It will occur with or without oxygen. If oxygen is not present, it will continue on into fermentation. If there is oxygen, we will find the oxygen inside our mitochondrion and allowing cellular respiration to continue. The second step is the transition step. It is here that oxygen must be present in order for us to continue. What happens is the pyruvate created at the end of glycolysis is then taken into the mitochondrion. As it goes across the membranes of the mitochondria, it will reduce itself to acetyl coenzyme A carbon dioxide molecule, and another NADH plus H plus is reduced. So a 3-carbon pyruvate molecule loses a carbon in the form of carbon dioxide as it goes across the membranes of the mitochondrion. Acetyl-CoA is a 2-carbon molecule that in turn has a coenzyme attached to it. This transition step takes pyruvate, breaks it down by releasing another carbon dioxide and creating more NADH. Remember, NADH are our electron carriers. They are like a taxi cab driver. The more fares you pick up as a taxi cab driver, the more money you can produce. NADH is the same way. The more NADH that can be made throughout the process of cellular respiration, the more energy can be produced. Because there are 2 pyruvates created at the end of glycolysis, we do see this transition step occurring twice. The third step is the citric acid cycle. The citric acid cycle takes place inside the mitochondrion. It actually occurs even deep inside the mitochondrion at the matrix. What is going on here is we take this now 2-carbon molecule of acetyl-CoA and release 2 more carbon dioxide molecules. In the process, the hydrogens and electrons are released, allowing for oxidation reactions to occur. We do see 3 NADHs reduced, and that other coenzyme, FAD, reduced as well. For each turn of the citric acid cycle, we have 2 carbon dioxides, 3 NADHs, and 1 FADH2. But recall, there are 2 pyruvates, and so this cycle has to occur twice for 1 glucose molecule. So to recap, we have glycolysis occurring for step 1 in the cytosol. Step 2 occurs as the pyruvate goes across the membranes of the mitochondrion, and step 3 occurs inside the matrix of the mitochondrion. The last step of cellular respiration is the electron transport chain and oxidative phosphorylation. The electron transport chain occurs inside the mitochondrion as well. It is at this point where we are going to see the bulk of all of our energy produced. Recall that during glycolysis, the transition step, and the citric acid cycle that we reduced NADH. For each NADH that we reduced, it is in the electron transport chain that we see it come to fruition. Through a series of oxidation and reduction reactions, each NADH is able to produce 3 ATP molecules. We also see each FADH2 made being able to be converted to 2 ATP molecules. The process of making these ATPs is what's known as the chemiosmotic theory. Chemiosmosis is the process of allowing electrons to travel down a series of proteins, and it is this flow of electrons that actually allow for the production of ATP to occur. As you can see here, this third step actually occurs, or fourth step, my apologies, actually occurs in the membranes of the mitochondria. Chemiosmosis, don't let it confuse you with osmosis. It is not the diffusion of water. It is a chemical gradient that is built up by hydrogen ions released through the oxidation of NADH. That is the driving force for the production of ATP. This ATP is made by what's known as oxidative phosphorylation. If you seem confused, don't worry. As we go through this process of talking about each of these steps individually, it will start to make more and more sense. How do we actually make ATP? Well, there are 3 mechanisms to phosphorylate ATP. Remember, phosphorylation is the addition of a phosphate group to any kind of molecule. In the production of ATP, we are actually adding phosphate to ADP, or adenosine diphosphate. One way of producing ATP is through what's called substrate level phosphorylation. In substrate level phosphorylation, we see an ATP being produced from a substrate having a phosphate group attached to it and an ADP molecule. The two enter into an enzyme and come out with a formation of ATP. A second type of phosphorylation is oxidative phosphorylation. We will look at this in more detail as we talk about the electron transport chain. A third type of phosphorylation is photophosphorylation. This occurs in photosynthesis, and it is the input of light, or those photons, that is actually the driving force of photophosphorylation. Well, let's look at substrate level phosphorylation in a little bit of detail. In substrate level phosphorylation, there is a substrate, any substrate that has a phosphate group attached to it. This substrate will bind to the enzyme, and in another place, ADP will also bind. Remember, ADP is a lower form of energy than ATP. As the two bind to the enzyme, the phosphate group goes and is transferred from the substrate to ADP to create ATP. Another way of looking at this, as we see here, here's our molecule with a phosphate group attached to it. Here is ADP with 2 phosphate groups attached to it. Here is our enzyme with all of its binding sites. This substrate, in substrate level, will actually bind to the enzyme, and so will ADP. We see them binding to the enzyme, and then the chemical reaction of this bond breaking here and a new bond forming here will occur. When that happens, we now have ATP and our high energy molecule without any phosphate groups left over. This now is with a low energy molecule, and all that energy has now been transferred to form ATP. This occurs at the substrate level. This occurs on the substrate. So you have to recall that that is the only way that we will get this type of ATP being produced. Oxidative phosphorylation is a little bit more complex because it is not a tangible reaction. When energy is released from our food, if there was no way of trapping that energy, it would be one large explosive reaction, and a lot of the energy would be released as heat as well as light. But because electron transport chain and because cellular respiration needs to have some control, it allows for reactions to occur in baby steps. These baby steps allow us to remove our food or remove hydrogens and electrons away from our food sources and carry them to the electron transport chain from our energy shuttlers, NADH. Then NADH becomes oxidized back to NAD+, and the hydrogen ions and electrons that it was carrying will transfer down a chain of proteins, going through a series of redox reactions. NADH is oxidized because it loses an electron. Well, this first protein here then is reduced because it will gain that electron. Then this first protein is oxidized and will lose its electron to the second protein, which becomes reduced. And then we see him oxidized and the third one reduced, then this one oxidized, the fourth one reduced, this one oxidized, the fifth one reduced, this one oxidized, the sixth one reduced. Once he is oxidized, he will lose his electrons and become oxidized, and oxygen will pick it up and become reduced, and in process form water molecules. Well, as these electrons are jumping downhill, traveling downhill, they are actually producing ATP. So there's a huge difference between substrate level phosphorylation and oxidative phosphorylation. This is oxidative phosphorylation is happening because all these proteins are being oxidized, which started with the oxidation of glucose to begin with. Once again, as we move forward, this hopefully will make more and more sense.